Good afternoon and welcome to this webinar on the update of the Food Additive Guidance organized by the EFSA Food Ingredient and Packaging Unit. My name is Camilla Smeraldi and I will guide you through this event that is taking place today. And to start with, some housekeeping rules to keep a smooth and effective meeting. Kindly note that you are automatically audio broadcast. However, this webinar is only in a listening only mode, so there's no way that you can interact during the meeting. The event is taking place in English. There's no translation available. And this event is being recorded. And at the end of this meeting, the event will be available publicly on the EFSA website. Finally, after the event is over, you're kindly requested to fill in a survey, and to do that, you will receive a link to express any thought or feedback that you may have on this event. Just at a glance, we were glad to have a very warm reception to this event. So we had more than 400 registrants from 56 different countries. In terms of the type of stakeholders that we have reached with this event, as somehow expected, our main stakeholders are coming from the private sector. Of course, this document being the guidance that is applicable to the submission of food additive applications, but we were really pleased to see a wide range of different audience that we may reach with this event. So we hope it will be an interesting one. So what are the objectives of today's webinar? The main objectives of today is really to explain you the content and the main changes that are being implemented in this updated guidance document. We would like also to make sure that the process of public consultation, which is currently ongoing, is clear and that we can illustrate it to all the participants. And this is really to stimulate the submission of comments and achieving a very good level of consultation with all the stakeholders that can contribute to the finalization of this draft output. During today's webinar, we will also start addressing the question and the request for clarification that we have already received from the moment that this draft output is already in public consultation. So I won't be alone, but I will be accompanied by my colleagues from the Food Additives and Flavorings team that have been supporting the development of this updated guidance. They cover different areas of expertise. So together with me are today Carla Martino, Panagiotta Zakidou, Alexandra Tard, Stefania Barmaz, and Laura Ruggeri. You will hear them intervening at different stages in the presentation, addressing the different sections of the guidance which is under revision. So we started on time, 2:30, with this opening of the event. And then we will go through the update of the guidance with a focusing on why we are updating the guidance and what is really the content and the proposed changes. We plan to take a short break of approximately 10 minutes, which we estimate could happen at around 3:30. And then we will resume after the coffee break with a question and answer session that will address the questions that have been received during this initial phase of the public consultation. The questions were not as many as we had planned when we scheduled the event, so it's likely that the closing time will be earlier than what originally announced. Finally, yes, I will wrap up on the content of the event, the next steps, and we will say bye. And with that, I will start with handing over the floor to my colleague Carla Martino, that she will really guide you through the why, so the background to this update of the guidance and the general principles that have somehow steered the current ongoing revisions.
Thank you, Camilla. Good afternoon, everyone. Thank you for joining this webinar. So together with my colleagues, I will guide you through the update of the Food Additive Guidance, highlighting the proposed changes compared to the currently applicable guidance that was issued in 2012, and since then it never underwent a scientific update. Next. So in the course of the fourth quarter of 2023, the EFSA panel on food additive and flavorings identified already the need to revise the 2012 guidance for submission of food additive evaluations. And in November 2023, a self-task mandate was approved by the EFSA Executive Director. The mandate is published on OpenEFSA and available at the link on the slide. In the term of reference as provided in the EFSA self-task mandate, it is mentioned that in accordance with Article 29 of the founding regulation of EFSA, EFSA is requesting its FAF panel to update the guidance to take into account the experience gained with the practical implementation of the 2012 panel guidance in the assessment of food additive applications submitted under Regulation 1331/2008. And where possible, the panel should ensure consistencies with applicable horizontal guidance documents, as well as with the latest guidance for the risk assessment on food flavorings that was adopted by the FAF panel in 2022. Next one, please. So during the course of 2024, we have been working actively with the experts of the FAF panel to update the content of the guidance, and really the first focus was to account on the experience gained with the assessment of food additive applications received in the latest years, and to clarify all the issues that have been subject to additional data requests issued by EFSA to applicants to submit and ask applicants to submit additional data. So the aim is really to clarify these issues in the guidance, to make clear the data requirements, and to avoid the submission of incomplete technical dossiers, to avoid to stop the clock and to interrupt the risk assessment. Another focus was really to update the reference to any horizontal and cross-cutting guidance documents, namely the EFSA Scientific Committee guidance documents. Some of them were obsolete or missing, and also to implement any development in science and in risk assessment methodologies. Also, we put attention to align the content with the recent sector-specific guidance documents that EFSA issued on regulated products with elements in common to food additives, so not only food flavorings as mentioned earlier, but also novel food, for which guidance has been recently updated and published in September last year. Next, please. So in terms of timelines, in November 2024, the draft Food Additive Guidance was endorsed by the FAF panel, and the public consultation was released just before Christmas, and it is planned to stay until 21st of February 2025. In between, we have organized this stakeholder webinar. And following the public consultation, then the adoption of the guidance is foreseen in June 2025, after having analyzed all the comments received through the public consultation and see if changes and updates are needed. A transition period is also foreseen at least 6 months before the implementation of the revised Food Additive Guidance. Next one. Here you have the link to the public consultation and a screenshot of what you will see on the EFSA website where the public consultation is available. You can download the PDF of the guidance from that site, and your comments needs to be submitted electronically by pressing the Submit Comments button. Next one, please. And now we really dive into the content of the guidance. So the main section of the revised Food Additive Guidance are listed on the screen. It is organized in 6 main sections. So an introduction, section 2, very brief information on existing authorizations and evaluations. Section 3, 4, and 5, and 6 reflects really the content of a technical dossier, so characterization and specifications, proposed uses and exposure assessment, toxicological data, and safety for the environment. I will guide you through the content of the first 2 sections, and then I will pass over to my colleagues for the remaining sections. Next one, please. So starting with the introduction, the section starts with the chapter scope, in which really the scope of the guidance is explained. So the draft guidance describes the type and quality of the data that are needed from the applicant to assess whether the proposed food additive, meaning the food additive for which an application is submitted, is safe under the proposed conditions of use. It applies to applications submitted under the so-common Common Authorization Procedure, Regulation 1331/2008, and it applies to authorizations for a new food additive or to modifications of an already authorized food additive, which may be related to changes in the manufacturing process, its specifications, or its conditions of use. Any procedural or administrative aspects linked to the submission of an application fall outside the scope of this guidance, which includes only a scientific update, and this information, on the other hand, can be found in another guidance, EFSA guidance, the administrative guidance on food improvement agent, that is planned to be updated in parallel to the scientific guidance. Next one, please. So the following section is, under introduction, gives some general principles that should be read before entering into the data requirements. This on the slide is not an exhaustive list, it's just a selection of the most relevant principles. So the first one refers to the EFSA horizontal scientific guidance documents that are mentioned, cited throughout the document. Most of them refers to the EFSA scientific guidance documents. And applicants should follow the latest available versions of these documents when compiling the technical dossier, and any newly available guidance relevant for the evaluation of the food additives, which means that the version of the guidance that is cited in this document could not be the latest one. In fact, the update of the guidance maybe happens in a time when there is an update ongoing of one of the scientific committee guidance, and the applicants are requested to scrutinize this update and really to refer to the latest available version. The next one refers to the minimum data requirements. So data on identity, manufacturing process, specifications, proposed uses and use levels and exposure represent the minimum data requirements which must be fulfilled in all applications for a new food additive. Toxicokinetic and toxicity data should also be provided unless the applicant can provide scientific justification as to why new data are not needed, because they are available from the public literature, for instance. Concerning re-evaluation of food additives that are submitted according to Regulation 257/2010, These applications falls outside the scope of this guidance. However, the principle of the guidance should be considered by interested business operators also when submitted the requested data from the calls for data in the context of the re-evaluation process. Next slide, please. Always under introduction, there is a chapter referred to the risk assessment process. It's the only section of the guidance where EFSA explains how the risk assessment will be conducted by the FAF panel. In fact, the other sections are more focused on the data requirements needed to be considered by the applicant to generate the data to be included in the technical dossier, whereas this part of the guidance try to explain briefly how EFSA will conduct the risk assessment. That indeed is divided in the 4 steps, in the classical 4 steps of risk assessment, starting with the hazard identification. So the identification of any potential adverse health effects derived from the use of the proposed food additive based on the results obtained from toxicological studies. Then it moves to the hazard characterizations, which consist in the evaluation of the dose-response relationship for the most sensitive adverse health effects, and the identification of a reference point used to derive health-based guidance value, which in the case of food additive consists in an acceptable daily intake, an ADI. Or if not appropriate to derive the ADI because the toxicological data set is insufficient, then it could be used, the reference point, to calculate a margin of exposure, which is the ratio between the reference point and the estimated exposure. And in fact, the next step is actually the assessment of exposure, which is the combination of the food consumption data from the EU national surveys with the proposed use levels of the food additive as submitted in the technical dossier. And it ends with the risk characterization, which is the evaluation of the health risk in the exposed population made by the comparison of the highest exposure estimates, the P95 percentiles of the exposure, with the ADI, or if the ADI cannot be derived, cannot be set, then it consists in consideration of the magnitude of the margin of exposure, which should not indicate a safety concern for the proposed food additive. Next, please. There is also a section explaining how EFSA would perform the risk assessment of impurities present in the proposed food additives and indicated in the specifications of the additives. With the word impurities, the guidance refers to any residuals and byproducts resulting from the manufacturing process and of the proposed food additives. So there are 4 categories of situations, let's say, where the impurities can be assessed in a different way, depending on the data available on the impurities. So for impurities for which toxicity data are not available, then non-testing method like read-across, SAR analysis or TTC approach may be used for a preliminary assessment of their toxicological potential. For impurities instead for which data are available and for which, for example, a reference point or the HPGV can be set or derived, then the potential exposure to these impurities from the use of the proposed food additive is calculated. And this exposure is then compared with the available reference point or HPGV for the impurity. Another situation is when impurities are present that are both genotoxic and carcinogenic. In this case, a marginal exposure approach is applied in line with the scientific committee guidance from 2012. Limits of such impurities in the specification should be as low as possible and should result in a marginal exposure of at least 10,000. Examples of the risk assessment of impurities falling under the 2nd and 3rd bullet point on the slide are available in the EFSA opinion published in the recent years when it comes, for example, to the risk assessment of toxic elements such as lead, cadmium, mercury, or arsenic that really falls under these situations. Then the last bullet refers to impurities that are genotoxic, but for which no carcinogenicity data are available. In that case, a TTC approach is considered in line with the Scientific Committee guidance from 2019. So in this case, it will be checked if the exposure estimates based on the proposed maximum use levels are below the TTC for genotoxic compounds of 0.15 micron per person per day. Next one, please. So the section on the introduction section is closed, and now we move to the second section. It's a very short section in the guidance, which requires applicants to inform about existing authorizations and evaluations. So information on these existing evaluations should be included in the technical dossier, if applicable, and details on the regulatory bodies that carried out the evaluation should be provided, together with the time of the evaluation and any relevant data and studies conducted in that context, together also with the outcome of the evaluation and if any reference points or health-based guidance values have been derived. This was my last slide for the moment, so I now pass the floor to my colleague Panagiotta, who will describe the next section of the guidance.
Thank you, Carla, and good afternoon to all. The next section is the characterization and specification section, which highlights the main parameters needed to fully characterize and specify the proposed food additive. It consists of 6 different subsections, which we will see one by one in the next slides. In the first subsection, named General Information on the Requested Data to Support the Identity and Specification, we can see that this is a newly introduced subsection which intends to address recurring questions raised in the ADR letters by our working group experts regarding specific technical data. We have 3 different bullet points or paragraphs. The first one focusing on the batch-to-batch variability and the number of batches to be provided. It is mentioned that the analytical data on at least 5 independently produced batches should be provided to demonstrate that the food additive can be consistently manufactured within the proposed specification, and of course for a specific method of manufacturing. The next paragraph refers on the analytical methods used and their sensitivity and specificity. It is mentioned that the analytical methods should be applying state-of-art techniques and be sensitive, specific, and fit for purpose, and information on the limit of detection, the limit of quantification, and other validation parameters should be provided in all cases. And the last one refers to analytical results data to be provided, that they should be supported by adequate certificates of analysis, which can be internal or from external laboratories. And in all cases, the analytical data should be provided as numerical values and not reporting limits that are different from the LOQ or the LOD. In the next slide, we discussed on the identity of the proposed food additive. Here we can see that the categories were consolidated and merged in order to streamline information. We have 4 different categories, the first one being single substances, simple mixtures, meaning mixtures the components of which can be fully identified, and polymers. The second category consists of complex mixtures, meaning mixtures the components of which cannot be fully characterized and a portion of unidentified components can be found. And this can derive from plants, macroscopic fungi, macroalgae, animals, or their parts. The next discusses on substances containing or derived from microorganisms, and the last, engineered nanomaterials. It is important to underline that there may be cases where 2 or more subsections can be of relevance to the proposed food additive, and in that case, information from all those subsections should be provided. The analytical data should be provided, if we see in the previous slide, for the food additive without the inclusion of carriers or diluents and on a solvent-free basis. And for the third and fourth category, it is important to mention that the cross-cutting guidance documents on microorganisms and nanomaterials have been added. In the next slide, we see the data needed in general to be provided in this identity section. The first one being the purity assay value of the substance, or in case of mixtures, the functional component, meaning the component that provides the technological function of the proposed food additive. Then the concentration of potential impurities, such as solvent residues, toxic elements, mycotoxins, polycyclic aromatic hydrocarbons. The concentration of major classes of components, such as carbohydrates or characteristic single components of the proposed food additive, like polyphenols or other components of potential concern in case of mixtures. The proportion of the unidentified fraction in case of the complex mixtures. Chemical characterization information for the chemical names, identification numbers, stereochemistry or other common names. Physicochemical parameters such as the appearance of a solution or a solid, the solubility particle size if relevant, the pH, and for the second category, the origin, parts used, and growth conditions. In the next slide, we see the requirements needed in terms of manufacturing process, which can be generic and specific. For the generic requirements, a detailed description of the manufacturing process should be provided. Here it should be noted that information provided to the EFSA will be treated with confidentiality, and this is why details can be disclosed. The identity and purity of the raw materials and solvents used, some key process parameters and relevant steps, for example, an encapsulation step. Information on purification steps, important for the impurity and the different impurities, and drying methods. For example, a spray drying step could induce the formation of nanoparticles. And as supportive information, a production flowchart can be useful. For the specific requirements, we have divided 4 different sections, meaning the proposed food additives manufactured by chemical synthesis, in which information on the starting reagents, the reaction conditions, the catalyst use, and the purification methods should be provided. For proposed food additives manufactured by fermentation, information on the different fermentation steps and reactions, the system, and a list with the raw materials should be provided, and those are in line with the EFSA step panel guidance document on food enzymes of 2021. The same guidance documents is used for the proposed food additives manufactured by enzyme catalyzed synthesis, where information on the identity of the enzyme should be provided, its strain, the inactivation and removal, and the status of the enzyme assessment by EFSA. And the last category for proposed food additives derived from plants and macroscopic fungi, microalgae, and animal sources, where information on the procedures employed, the usually extraction steps, reagents, and the production yield. The next section refers to the specifications which define the requirements concerning the identity, the purity, and the limits of any impurity present in the proposed food additive. As for the identity, the information should be given without carriers or diluents and on a solvent-free basis. And the information to be included is a definition of the proposed food additive with information and details on the manufacturing process applied, such as which are the raw materials used, the different manufacturing steps, if enzymes, microorganisms, and their strain, solvents, and catalysts are used. Relevant parameters such as chemical name, chemical formula, molecular weight, solubility, and pH, the purity assay value of the substance or of the functional components. In the case of mixtures, the content of the major classes of components present and characteristic single components of the proposed food additives and other identified components, limits of impurities such as solvent residues, toxic elements, mycotoxins, and microbiological criteria. In the next slide, we can see that in the guidance document, Appendix 8 includes a format for the proposed specifications of food additive. It should be mentioned that this is a template table and it should be adapted by the applicant, who is the one knowing best what they intend to put on the market. And for that reason, further parameters not included in the table may be relevant and should be proposed by the applicant. In the next section and slide, we see information requested on the stability, reaction, and fate in food. And two different issues should be examined. The first one being the stability of the proposed food additive during storage in shelf-life and under accelerated storage conditions. Of the proposed food additive as produced, meaning as a solid or liquid, and of the proposed food additive in food or in relevant model system representing the intended conditions of use. Then, the reaction and fate of the proposed food additive in food, important for the different transformation and degradation products that could derive, and information on their identity, concentration, stability, and reactivity should be provided, along with the nature of interactions and reactions of the proposed food additive with components of food. The function of the food additive is examined in terms of risk management. This is why it is not mentioned in our guidance document. And in the next slide, the last subsection refers to the methods of analysis used to evaluate the proposed food additive in the specific foods it is intended to be added, and it emphasizes on the method's specificity, repeatability, reproducibility, and the fact that it should be fit for purpose. And with that, we conclude on the characterization and specification section, and I pass the floor to our colleague, Alexandra.
Thank you, Yoda. And now we are going to see the parts related to the information needed for the exposure assessment, meaning the proposed use and use levels, and the data exposure assessment scenario. So first, the proposed use and use level. Applicant in their dossier should indicate in which food or food category the food additive is proposed to be used. At least the food category should be provided according to the food nomenclature described in the Annex 2 Part D of the regulation number 1333/2008, and this is mandatory. It is also recommended that the Food X2 classification system is used. Indeed, this food classification system, which is developed by EFSA, allows to give more detail on the foods. In any case, the more information is provided on the food, which is requested for containing the proposed food additive, the best it is. For each of these foods, then, two proposed use levels should be provided. The maximum use level, which would be the maximum level at which the proposed additive is expected to be used in that food, and the typical use level, which is the common use level at which the additive is expected to be used in the food. This level should be expressed in the food additive itself, in the case if it is a single substance, or in the functional component, in the case the food additive consists of a mixture. Next, please. Added to that, the information on the concentration of the proposed food additive is needed if carryover of the proposed food additive is expected from its use in food additive preparation, flavoring, enzyme, and/or nutrient, or if the proposed food additive is to be used in food used as ingredient in composite dishes. Next, please. With this information on the use level, the applicant is requested to assess the dietary exposure to the food additive, and this should be done with two EFSA dietary exposure tools, namely FAME and Diatext. FAME follows the additive legislation, and the food category in that tool are the one described in this Annex 2 Part D of Regulation 1333/2008, while the Diatext tool follows the nomenclature of the Food X. Both tools are based on data from the EFSA Comprehensive European Food Consumption Database. And this tool can assess the dietary exposure for the general population, meaning from infants from 16 weeks of age to the elderly. Submitting exposure estimates based on FAME is mandatory, while using Diatex is recommended. In the following slide, we will see which scenario should be performed with the proposed use level and these tools. So first is the case of a new proposed food additive. This is the simpler case. All the proposed food additives should be all the proposed food categories, should be selected, introducing for each of them the proposed maximum use level as for a first scenario, and then the proposed typical use level for a second scenario. This means two exposure estimates. This means two exposure estimates when using the FAME tool. And if the applicant uses also the Diatex tool, then there will be again two exposure estimates, one with the maximum and one with the typical proposed use level. This will lead then to 4 exposure estimates. Food supplements and food for special medical purposes for infants and toddlers should not be introduced in this tool, as they will be considered in other scenarios that we will see later on. The second case is when the food additive is already authorized, or and if the application is for authorizing it in new food category, or is for authorizing in new food category or in food category to be modified, requested to be modified. So in the first step for the new food category or the food category requested to be modified, this food category should be selected in the tool and attributed the proposed maximum use levels. Then, for the already authorized food category, meaning those for which no modification is requested, they should be selected in the tool and attributed their MPL in the case there is a numerical MPL. In the case there is no numerical MPL, for that food category and that additive, the applicant should consider the maximum concentration level taken from the last EFSA opinion. If such an EFSA opinion is not available, or if no concentration has been reported in that opinion, then the food category cannot be considered in the exposure assessment. This will provide the exposure estimates according to scenario maximum 1. For a second scenario, the applicant should keep the proposed maximum use level for the new or the requested to be modified food category, while for the food category already authorized, for which no modification is requested, the applicant should use the mean of the concentration levels used in the last available of EFSA opinion, and this will provide estimates according to scenario maximum 2. Next, please. The same scenario should be provided by using the proposed typical use level for the new proposed food category or food category which use level is requested to be modified. And this will lead to the scenario typical 1 and typical 2 respectively, using the maximum or the MPL or the maximum use level or the mean use levels from the last EFSA opinion. Thus, if exposures, if there is no EFSA opinion on the same food additive with reported concentration level, then the scenario maximum 2 and maximum typical 2 cannot be performed. In the following slide, you will see the flow chart which is also in the guidance documents, which summarize what was presented in the 2 previous slides. In the left side, you can see the side on the new food category or food category for which a modification is requested. On the right side, the food category for which a modification is not requested. And in the middle, the resulting part or the resulting exposure scenario. All the results, as well as the concentration value used, inputted in the tools, should be provided in the dossier in an Excel file. So we mentioned earlier that the tools that are used for estimating the exposure to the general population should not contain the food supplements and food for special medical purposes for toddlers and infants, because they are considered in other scenarios that we're going to see now. So for the infants below 16 weeks of age, the exposure assessments follow the guidance of the Scientific Committee of EFSA, which was published in 2017. And this guidance document provides mean and high-level consumption amount of infant formula for assessing the exposure to substance which do not accumulate in the body. This recommended value for this substance, like additives, are 200 milliliter at the mean and 260 milliliter per kilogram body weight per day at the high consumption. The exposure for infants below 16 weeks of age should be calculated for the different food category corresponding to food for infants, for these infants, meaning the standard infant formula which is the food category 13.1.1, and the infant formula for special medical purposes, which is the food category 13.1.5.1. If the applicant should use the conservative mean and high-level consumption amount of infant formula combined with the proposed maximum and typical use level of each infant formula to calculate the dietary exposure to the proposed food additive in infants below 16 weeks of age. In the following slide, we're going to see when the proposed additive is requested to be used in food supplements and or in dietary food for infants and young children. As for these two food category, a consumer-only scenario will be performed. However, this scenario cannot be performed via the available tool made available by EFSA, and therefore we will have to perform them. In the case, however, the proposed food additive would be proposed to be used only in food supplement, the applicant can and should estimate it with the use of the Diatext tool, because this tool allows to calculate the exposure on food consumers only, considering only one food. The guidance also indicates that an acute exposure assessment will be performed by EFSA if relevant, meaning in the case of acute effects of the proposed additive. Next slide, please. Considering that a proposed food additive can also be a substance present in food for other reasons, it is also requested from the applicant to provide information on the use of this substance from other dietary source or from non-dietary source. In the case of dietary source, the proposed food additive could be naturally present in the food or added to food as flavoring or ingredients, or present in the food due to its use in food contact material or plant protection product. Regarding the non-dietary source, the proposed food additive may also be used in medicine, cosmetics, or tobacco products. The information requested is qualitative, so meaning concentration level, consumption amount, estimates of exposure, and if this is not possible, qualitative information from literature, for instance, or database should be provided. Based on all these exposure results, the exposure to any toxicologically relevant impurities should be estimated also by the applicant pro rata from the exposure to the proposed additive. So this is all concerning the overview of the exposure scenario of the exposure section of the guidance. I will now pass the floor to my colleague Carla for the next part of the presentation.
Thank you, Alexandra. So now I'm back to present you the content of the section 5 of the updated guidance, toxicological data. So this section starts with a list of issues that needs to be considered before or when conducting toxicological studies. Again, the list here is not exhaustive, it's just a selection of the most relevant ones. So first of all, toxicological studies should be carried out with the test material manufactured according to the manufacturing process specified in the technical dossier and in compliance with the proposed specifications. Regarding the literature search, a comprehensive search should be carried out before embarking in any toxicological studies, and the literature search should cover all the toxicity endpoints described in the guidance, including the safety for the environment. This, in fact, is in line with the next point, so try to avoid any unnecessary animal studies, in line with the 3R principles of reducing, refining the unnecessary animal studies, in line with the EU directive from 2010. And in this respect, also new approach methodologies can be used as alternative methods to demonstrate the safety of the proposed food additives. These methodologies should be validated and standardized by an official bodies, for example, OECD or JRC, to be used for regulatory purposes. If they are not validated or standardized, they could be otherwise used as supporting evidence. For botanicals that are used as food additives, a presumption of safety may be applied when they are derived from conventional food sources with a long-term history of safe use, and if data on exposure show that no significant increase of intake compared to historical levels is to be expected due to the intended use levels. And this is in line with the principles of the Scientific Committee Guidance from 2009 on botanicals. Regarding read-across, it is mentioned that it may provide a possibility to avoid, again, unnecessary toxicity testing. If read-across is applied, the general provisions outlined in the OECD and ECHA guidance documents on read-across should be followed. In this respect, it is worth mentioning that EFSA is developing a guidance on the read-across. So when it will be available, also the principle of this guidance needs to be considered and followed. Next one, please. So the section 5 continues with this chapter on safety evaluation and corresponding testing strategy. This is a new section that was not present in the 2012 guidance. It's an important section that is reflected also in the electronic submission tool, where the applicant is requested to justify the testing strategy used to demonstrate the safety of the proposed food additive. And this includes the rationale for inclusion and exclusion of specific in vitro and in vivo toxicity studies. Also, in this section, applicants need to investigate and provide information regarding the potential presence of small particles, including nanoparticles, in the proposed food additive. And in case the food additive contains nanoparticles, then the horizontal EFSA Scientific Committee Guidance on technical requirements to establish the presence of small particles, including nanoparticles, should be considered as a starting point to decide the strategy for the risk assessment to follow. So in particular, if the conventional risk assessment is sufficient or not. If it's sufficient, then the data requirements can follow the sectorial guidance, meaning the food additive guidance. But if the conventional risk assessment is not sufficient due to the presence of nanoparticles, then the assessment needs to be complemented by additional studies or considerations in line with the principles of the Scientific Committee Guidance on nanomaterials. Next. The next issue tackled in the toxicity section refers to the tiered approach for conducting toxicokinetic and toxicological studies. This really is the core of the toxicological data section. So the tiered approach recommends the conduction of animal studies to be conducted up to a point where the information is enough to conclude on the safety. It is divided in 4 tiers. In each tier, number of studies are recommended, and the higher the tiers, and the more complex and demanding would be the toxicity testing. One of the changes compared to the 2012 guidance is really this tier 0, which encompasses genotoxicity testing, which should be conducted before embarking in any other in vivo toxicity testing foreseen in the other tiers. The color code is really genotoxicity testing in green, toxicokinetic in yellow, and repeated dose toxicity testing in blue, encompassing subchronic toxicity, developmental and reproductive toxicity, chronic toxicity, and carcinogenicity. Next one. So why in tier 0 we define this tier 0 genotoxicity assessment? So the assessment of genotoxicity potential of a food additive should be carried out before embarking in any other in vivo toxicity studies as mentioned earlier, other than to test for in vivo genotoxicity or to study target tissue exposure. A stepwise approach is to be followed for the generation and evaluation of genotoxicity, as it is described by the EFSA Scientific Committee Guidance on Genotoxicity. At the moment there are 4, and EFSA is currently revising these guidance documents and to issue a single document. So genotoxicity is an endpoint per se, so a clear evidence of genotoxicity in somatic cells in vivo has to be considered an adverse effect per se, even if the results of carcinogenicity study are negative, since genotoxicity may be also implicated in other diseases other than cancer. Therefore, if the proposed food additive is concluded to be genotoxic in vivo via a relevant route of administration, there is a concern for genotoxicity for the proposed food additive, and no further toxicity testing should be performed. So this is why the genotoxicity assessment was presented in this tier 0. Next slide, please. So regarding the genotoxicity assessment, as mentioned, the strategy described by the scientific committee applies fully. So it is a stepwise. In step 1, in vitro genotoxicity testing are foreseen and consist in the bacterial reverse mutation assay and an in vitro micronucleus assay. If positive results are observed in these 2 tests in vitro, then an in vivo follow-up is needed, and one or more than one in vivo genotoxicity testing would need to be conducted. So you see in blue this mammalian erythrocyte PIGA gene mutation assay, which is an in vivo genotoxicity testing that is applied to follow up to positive results in vitro to gene mutations. This study, the OECD test guideline 470 describing this test, was not available at the time of the development of the testing strategy by the scientific committee, and so it was not included by the scientific committee in the respective guidance, but now it is available, and we have included in the updated guidance on the draft guidance of food additive. Next one, please. So if the food additive consists of mixtures, then the genotoxicity assessment follows the recommendations, again provided by the scientific committee in the statement on genotoxicity assessment of chemical mixtures from 2019. So food additive consisting of mixtures may be either fully defined mixtures, the so-called simple mixtures, or complex mixtures, which means mixtures containing a substantial fraction of unidentified component. So in both cases, the components of the mixture should be identified and quantified as much as possible. And conclusions on genotoxicity are required for all identified components, so the identified components should be assessed individually with respect to their genotoxic potential. And if it is concluded that the mixture contains one or more components that have been assessed to be genotoxic in vivo via the oral route, via relevant route of administration, then the food additive raises a concern for genotoxicity. Next one. In case of complex mixtures, so when there is a fraction of unidentified components, only if none of the identified chemical substances in the food additive mixture raises a concern for genotoxicity, then also the fraction of unidentified components should be tested for genotoxicity. If it's not possible to isolate it, then the testing of the whole mixture should be undertaken. Again, the testing of the whole mixture follows the same genotoxicity testing strategy as described previously for single substances. And the draft revised guidance on food additive includes also additional recommendations regarding the interpretation of in vivo genotoxicity testing of these complex mixtures. And concern in particular the maximum doses to be used when testing these all mixtures, and the demonstration of target tissue exposures, which constitutes often a limitation in the interpretation of negative results from these in vivo genotoxicity testing. Next slide, please. And now it comes to the toxicokinetic and toxicity other than genotoxicity testing, of course, to be recommended to be conducted only if there is no concern for genotoxicity for the proposed food additive. These testings are described in the different tiers. I try to guide you through the decision tree, let's say, to move from one tier to the others. So the first recommendation is to start in tier 1 with the absorption studies. So studies to investigate the absorption of the proposed food additive and of its breakdown product via the gastrointestinal tract. So if absorption can be demonstrated, it means that the food additive is systemically available. These studies could be either in vitro, ex vivo, or in vivo, provided that the absorption can be investigated. Regarding the toxicity studies, a subchronic 90-day oral toxicity study conducted according to OECD 408 is recommended in tier 1. The study has the aim to assess whether any adverse health effects relevant for humans are identified in animal studies after prolonged and repeated administration of the substance to animals. The study can provide limited information on immunotoxic, neurotoxic, endocrine-mediated effects. Also, it can inform about effects on the reproductive organs with limited information, but it cannot provide any information on developmental toxicity. So for this reason, if absorption of the food additive or breakdown products is shown, is demonstrated, then in tier 1, the recommendation is to conduct a screening test for reproduction and developmental toxicity according to the OECD test guidelines 422, precisely to further investigate any potential effect on the reproductive organs and developmental toxicity in the prenatal animals. Again, if the absorption is shown, this would be the trigger also to move to tier 2 toxicokinetic testing. So we move at the center of the tier. So in this case, then it is recommended to conduct in vivo studies to investigate ADME, so absorption, distribution, metabolism, and excretion. following single or repeated dose administration of the substance. Guidance to in vivo ADME studies can be provided by the OECD test guidelines 417 using either radiolabeled or unlabeled substances. Another important element in Tier 2 toxicokinetic is the comparison of the metabolites that are relevant for human risk assessment. So the recommendation included in the EFSA opinion from the PPR panel on in vitro comparative metabolism studies can be followed. These are described in Appendix C of that opinion. And this will help in identifying whether any metabolites, human metabolites in vitro are similar to those generated in the in vivo ADME studies in animals. Regarding the toxicity study, the triggers to move to Tier 2 toxicity testing are represented by evidence of reproductive, developmental, neurotoxic or immunotoxic effects observed either in the OECD 422 or in the 90-day study. In this case, then, the recommendation is to conduct in Tier 2 an extending one generation reproductive toxicity study performed in accordance with the OECD 443. On the other hand, if no effects on developmental and reproductive toxicity is observed, but other effects in the 90-day study that are suggestive of tumorigenicity are observed, then the recommendation in Tier 2 is to conduct a combined chronic toxicity and carcinogenicity study in line with the OECD test guidelines 453. Regarding the engagement in Tier 3 testing, this should be considered with carefully. We also noticed in the guidance it is mentioned that based on our experience, the need to move to Tier 3 testing has been never identified based on the recent evaluations on the food additive. Anyway, one possible reason to move to Tier 3 could be evidence of bioaccumulation coming from in vivo ADME studies, for instance, and evidence of long elimination half-life. And in this case, the need is to identify whether this effect, this observation is relevant also to human. So any ADME studies available in humans, volunteers, or already available from the literature could help in assessing this relevance for humans. The same applies for toxicity studies, and let's say the specialized studies could be conducted in Tier 3 to clarify the mode of action and the human relevance to adverse effects that have been observed in lower tiers, Tier 1 and Tier 2 in animal studies. Next slide, please. So in case the authorization of the proposed food additives is requested also to be used in foods for infants below 16 weeks of age, then additional toxicity studies are needed compared to the one just presented, as recommended by the relevant scientific committee guidance for this population group. More specifically, specific studies in neonatal animals, for example, piglets, are required if the proposed food additive is not absorbed and not systemically available, and therefore its evaluation remains in Tier 1. So in this case, additional neonatal animals would be studied in neonatal animals would be required in addition to Tier 1 testing. If there is evidence of absorption and of systemic availability, then we saw that we move to Tier 2 testing, and indeed an extending one generation reproductive toxicity study will be conducted, but it should be performed in line with the recommendations of the scientific committee guidance, in particular with respect to the number of cohorts of F1 animals, so the 3 cohorts will be needed, and the need to perform a direct dosing of the neonatal animals as soon as possible after birth. The draft guidance cites also some examples of the EFSA opinion on food additives where the safety of the products used in foods for infants below 16 weeks of age was addressed. This was my last slide, and I'll pass the floor to Stefania for the last part of the presentation.
Thanks a lot, Carla. Good afternoon to everybody. So according to Regulation 1333 of 2008, so the Regulation 1333/2008 laid down rule to ensure an high protection of human health, taking into account where appropriate also the protection of the environment. For this reason, and in similarity with what was done previously in the case of flavoring and smoke flavoring, a section on the safety for the environment was added to the new guidance. The emission of food additive and their possible metabolite or breakdown product into the environment can occur via different routes. We can have the human consumption of food containing the food additive and then the emission to the environment, or we can have emission to the environment following industrial emission. The focus of the guidance is on the first route, so basically on the emission following human consumption, because the industrial emission is covered by specific regulatory framework. I have reported in the picture of the slide, because sorry, a schematic view on how food additive can reach the environment. So we have the food consumption of food containing the specific food additive. Then we have metabolism in human and excretion of the food additive as such and or the related product, so breakdown product or metabolite. Those substances reach the sewage treatment plant where they are biodegraded to a certain extent. And the substances which are not fully biodegraded can reach the environment, so water, sediment, soil, and groundwater. Next slide, please. Considering the specific emission route to the environment, according to the guidance, the decision on whether an environmental risk assessment is needed should be made on a case-by-case basis. Two main situations are identified. So in the case of food additive or its components which are reported to occur naturally in food or beverage which are consumed in the EU, normally the need to perform an environmental risk assessment is not anticipated. Nevertheless, in this case, the applicant should always conduct a comprehensive literature search. In the case instead of food additive or their components that are not reported to occur naturally in food and beverage consumed in the EU, further assessment is needed if one or more conditions are met. The first condition is that the proposed food additive is not extensively metabolized in human to innocuous product. It may be, in the case specific information is not available, that the metabolism information on metabolite and breakdown product need to be estimated. Some information can be anyway available from the ADME data. The second condition is that there is evidence that the proposed food additive and or its related product are persistent in the environment and, importantly, expected not to be fully degraded in a sewage treatment plant, and or they are expected to bioconcentrate. The last condition is that there is evidence retrieved in the scientific literature that indicate a potential environmental concern for the proposed food additive. So if one or more of these conditions are met, it may and further assessment for the environment may be needed. Next slide, please. In case of food additive or breakdown product or metabolite of the food additive are identified as persistent, bioaccumulative or toxic, or very persistent or very bioaccumulative, they will raise a concern for the environment, as in this case a safe concentration in the environment cannot be established with a sufficient reliability. In the cases where an environmental risk assessment is required, considering that there is no sectorial guidance available at the moment, what is recommended in the guidance is to follow the available guidance or guideline, which are available on the environmental risk assessment for substances which have a similar release pattern or exposure route. We have reported in here the recent guideline on the environmental risk assessment for medicinal products from EMA. Further information can be also obtained for other guidance, like the guidance on environmental risk assessment for biocide or industrial substances from ECA. This is the last slide on the presentation of the food additive guidance. We will now break for 10 minutes, but please do not disconnect because there is more to come. So thank you and see you later.
So thank you for being back with us after the coffee break, and thanks to the colleagues for having guided us through the content of the guidance. In the second part of the webinar, we will try and address the first comments that have been received during the registration phase as well to this event, as well as some of the comments that have already reached us during the public consultation. So, just as a reminder, the public consultation Carla mentioned earlier has already been launched, so the draft document is available online, and the consultation will close on the 21st of February. But at the time of announcing this event, the Today webinar event, there was also the possibility of submitting questions and comments to shape the content of this event at the time of registration. We received 13 questions that we will address today during this part of the webinar. However, also all the questions that, including those that have been submitted in the registration phase of this webinar, will also be addressed in writing because as it's common practice in EFSA, whenever we launch a public consultation in parallel to the publication of the updated document, we also publish a technical report with the outcome of the public consultation and how the different comments received have been addressed. So for the sake of clarity and streamlining also the flow of this event, we took the liberty of grouping the 13 questions that were received so that we could tackle them with a logic. So we start with some questions of generic nature, and I will address them. So the first one is concerning whether an additive can only be used in the listed food. So this question is probably not really in the scope of the guidance. However, as a general principle, we just take the opportunity to reinforce the concept that indeed, for a food additive to be lawfully used in the European Union, it has to be authorized, meaning included in the Union list, that's in Regulation 1333 from 2008, and also the same regulation specifies in which food categories and at which use levels the food additive can be used. So the content of the guidance that we have just presented is really the scientific content of a technical dossier that has to be submitted to accompany an application, so a request for an authorization of either a new food additive, so a substance which is not yet included in the Union list, so proposed for use as a new food additive, or for instance, a request for changing the already permitted conditions of use of a food additive. The second question was related to the fact if there were any changes in the submission steps from the past. What about additives that have already been approved and if an applicant is interested in extending their use? And again here, no changes to the application, and as I say, this is regulated under the Common Authorization Procedure for Food Additives, Flavorings and Enzymes. So the submission steps have not changed. Of course, over the years, there has been an evolution, and now submission of a dossier has to happen in an electronic format. So, and in this respect, we have tried to align the content of the guidance, also mirroring the content of the electronic tool that is used to submit an application. And the same also in the scope of the guidance, as we have explained, not only covers new substances that are proposed for use as new food additives, but also it covers those cases when an already approved food additive is requesting an authorization for different uses, or, for example, is requesting an amendment of the already authorized uses or changes to the specifications, for example, if produced from a different manufacturing process. The final question that I will tackle under those of generic nature is concerned the guidance on flavoring. So when the guidance for flavorings was published, it was emphasized that the requirements should largely align with food additives. This is now not the case, with significant difference in tox study requirements, and can we expect the flavoring guidance to be adapted accordingly? So to address this question, I would like to remind the audience that as we have explained at the very beginning, the mandate to update this guidance was requesting not only to align to the guidance for flavorings whenever possible, but more broadly to take into account any latest scientific developments. And we have mentioned, for instance, that, for example, in parallel to the update of our guidance, also the guidance for novel foods has been subject to a revision. And so the exercise conducted was really to reflect on all the experience that we have gained with the assessment of new application. Think about all the time that we had to go back to applicants with stop-the-clock letters, for instance, requesting additional data, either because then we thought that maybe the guidance, the existing one, was not clear enough or not covering a specific situation, but also because of the evolution in maybe horizontal guidance documents that were not available at the time of 2012. Having said that, there is also a procedural requirements that is dictated by the EFSA standard operating procedure that require guidance documents to be regularly revised. And again, among the elements to consider in the revision is really the practical experience gained. Now, with the guidance for flavorings, at the moment, we don't have any practical experience because we haven't received any dossier compiled according to the latest updated guidance. We have aligned as much as we could the two guidance whenever this has been possible, as it was mentioned earlier, for many parts related to the advances in horizontal guidance documents. And in other aspects, we have tried to align, and for sure this will be the reflection that we will make when time will arrive to revise the guidance for flavorings if any changes will have to be implemented. With that, I hand over to my colleague Jota for the questions on the characterization.
Thank you, Camilla. Regarding the characterization and specification section, we have received 3 different questions. The first one actually being more of a proposal. It is mentioned more specific on compositional analysis requirements for natural and fermentation-based products to be included in the guidance document. We would like to mention that a dedicated subsection has been included in the guidance document for proposed food additives manufactured by fermentation. And then regarding the natural-based products, these we understand that refers to the identity of the proposed food additive. For example, if it is a polymer, a complex mixtures derived from plants, and more information and analytical data should be provided. As already anticipated in the identity, there may be cases where 2 or more categories could be of relevance to the proposed food additives, and in those circumstances, the respective information should be provided. We have updated the guidance document based on the experience acquired over the years of the evaluation of the different dossiers. So it is really up to the applicant that best understands the identity and the manufacturing process of the proposed food additive to combine and provide all this information and data. Then, for the next question, it is mentioned what is the definition of significant change for the manufacturing process in food additives. For example, one gene edit is considered significant change in manufacturing process. Should the interested business operator or applicant notify or submit an assessment dossier to EFSA if there are changes in the microorganism used in the manufacturing, even though there is no change in the specifications? Now, a significant change of the manufacturing process is important because it can induce the formation of different impurities. In terms of our regulation of 1333 of 2008, for the authorized food additive, a modification, a significant change in the production method can be, for example, the additive that was previously produced from extraction from a plant to now production by fermentation using a microorganism, or the use of a conventional microorganism to a genetically modified microorganism. The modification of a starting material, for example, or the addition of an enzyme or an enzyme of a different strain, or the change in particle size, for example, a spray drying state that now can induce the formation of nanoparticles. Now, those shall be considered different additives. So for the already authorized food additives, the applicants should notify the risk managers, meaning the European Commission and the member states, and they are the ones to decide whether or not they require a new application and an evaluation by EFSA. Such an example can be the stevia glycosides. And then for any changes that take place during the risk assessment of the proposed food additive, because the applicant or the interested business operator maybe is a small startup company, those should be duly notified to EFSA, since, as we mentioned in the technical part of the dossier, the analytical data to be provided are for a specific method of manufacture. And the last question, number 6, do all listed food additives have a method of detection in the different matrices in which they are allowed? Now, in terms of risk assessment, which is the assessment EFSA works with, the information we request on the methods are the method of analysis in food of the proposed food additive, meaning we request a method that is sensitive, specific, and fit for purpose for the proposed food additive in the matrices it is intended to be added. The information mentioned in the question number 6 to have a specific method of analysis for the additive in the different food matrices really is more of relevance for the risk managers in order to monitor compliance with the specifications. And if such a list exists, no, there is not currently a list with the methods of analysis for each food additive and food category, but it will be considered. And with this, I conclude the questions on the section and I give the floor to my colleague, Alexandra.
Thank you, Jutta. The questions on the proposed use and the exposure assessment, they were 2, and they both relate to the scenario, to the consumer scenario. So the one point is to develop how EFSA is intended to prepare estimates based on consumer scenario, consumer scenario. And the second point is that considering the importance of for food additive producer to get an idea of the final estimates, and that the estimated consumer intake is a key parameter. Then how the applicant, since he cannot perform himself the consumer intake assessment, how can he have an idea of what will be the exposure results? So the consumer only scenario, in the consumer only scenario, the number of consumer depend on the foods which are selected. So if 2 foods are selected, the consumer will be, the people selected will be consumers of both food category or of one of these 2. So the consumer only scenario can be performed only with the access to the raw individual data. And as such, it cannot be done outside EFSA because only EFSA has access to this raw individual data from the survey which come from each member states. So estimates of the exposure to only consumers of food supplements, which is only one food category, can be done with the Diatex tool. It is indicated in the guidance also. This does not consider the whole diet, but this provide them information on the exposure coming from food supplements only. Then for the question related to the consumer-only scenario considering several food categories, this point is acknowledged. It will be also discussed internally in EFSA, and we will look into it to see whether another tool addressing this point can be developed. And for each of the application, then EFSA, for each of the application in which this scenario should be performed, so consumer-only scenario should be performed, EFSA will also engage in dialogue with the applicant to provide the information. And now I will pass the floor to my colleague Laura for the next questions.
Thank you, Alex. Now we cover the questions that are related to the part of the toxicological biological data. First of all, a question on the NAMs. So how the guidance promotes the use of the NAMs, and especially when these NAMs are not validated, how they can be used, or under which condition they can be accepted in the assessment. First of all, I would like to, let's say, state that the agency, so EFSA, promotes the use of new approach methodologies as an alternative of vertebrate testing. Of course, this is an area of continuous research and continuous development. So in the guidance, on purpose, we were not prescriptive in terms of method that can be accepted or not. In terms of, let's say, in the field of the regulatory science, of course, the preferred option is always to have a method that is validated to conduct the risk assessment. Nonetheless, the FAF panel is let's say, open to consider the NAMs, for example, that may be submitted by the applicant, even if validated. So the decision of the acceptability of non-validated NAMs will be taken by the panel on the case-by-case basis. Also considering, for example, other aspects, for example, the nature of the substance. Whenever, and this is a general, let's say, the general approach, whenever an applicant submits information according to methods or tests that deviates from what is recommended by the guidance, this can be done by the applicant, and the panel will assess this information, considering that duly justification should be given by the applicant. And of course, the justification for the deviation should be scientifically robust. Let's go to the second question, which is about the kinetic study. So the guideline, we see the guideline 417, when we have natural product and the radioenabling cannot be performed. Okay, first of all, when we have natural products, in many of the cases, we have mixtures, so we can consider them complex mixture or mixtures. In the case of the mixtures, first of all, it has to be considered whether there are components of concern, of toxicological concern in this mixture. And if there are, each component should be assessed independently. In case there are no components of toxicological concern, then the assessment should be done at, let's say, whole mixture level. And indeed, sometimes not all the test guidelines, even if conducted according to OECD, can be used in the mixture, in mixtures. So the indication here is for the applicant that knows best their product, that if they think that the OECD 417 is not suitable, then an alternative test can be provided as long as it's sufficiently sensible and suitable for the product. One note is that the OECD guideline 417 does not require always the radio labeling, so it is possible to conduct the study without radio labeling. Okay, then we have two questions related to mixtures, complex mixtures. First question is about a complex mixture and what fraction of the unidentified components will be considered substantial. And would that be based on the total fraction of unidentified components of the fraction of each individual component? Let me first say that the issue of the mixture, complex mixture, and this question is related to the genotoxicity assessment of the complex mixture. So the mixture toxicity, genotoxicity is let's say, well described. So the approach is well described in the EFSA Scientific Committee statement on genotoxicity assessment of chemical mixture, which is a guidance of the statement that was published in 2019. In the case of the complex mixture, this mixture contains a fraction of unidentified components. So not all the components have been chemically identified. Considering the wide, let's say, range of the mixture that can fall within this category, it was considered not possible to set a threshold or a cutoff value, so or a percentage of the unidentified components to consider these components, this fraction, be substantial. In the current guidance for the food additive evaluation, we provide some indication with regard to the elements that has to be clear and submitted by the applicant whenever there is a fraction of unidentified components. These elements you will find in the technical part of the guidance, and they are briefly, first of all, the proportion of the identified fraction should be provided, possibly in a quantitative amount on a weight-to-weight basis, and the analytical information to characterize the type of the components of the identified fraction should be also given. Next question is again on the genotoxicity assessment of the mixtures, and we have a question for the mixture and the complex mixture and identified components. So the question is, on what component or components would the maximum milligram-kilogram dose be based, and how would one determine what dose is high enough above the maximum limit to sufficiently increase the dose of each individual component? To reply to this question, I would start from defining the steps that one has to conduct to assess the genotoxicity of this complex mixture. So first of all the components has to be identified. So we have a mixture, several components. First step is to identify and quantify the components. The identified components should be then assessed individually based on all information available for the genotoxicity. If none of the identified components in the food additive mixture raise concern for the genotoxicity, then the genotoxicity potential of the unidentified fraction of the mixture should be assessed. Again, in case it is not possible or feasible, technologically feasible, to extract this unidentified fraction from the mixture and test it in isolation, the guidance recommends to test the mixture as a whole. Our guidance indicates some recommendation in terms of maximum dose to be tested. If there is no toxicity, so if there is no toxicity in other tests, then the guidance recommends to test at maximum dose that can be even higher than the limit dose that is indicated in the relevant OECD guideline. It could be a micronucleus or a cometacell. And of course, the highest dose has a limit, which is the maximum volume that can be administered to the rodents. The last question is about the margin of safety and why it is not considered in this guidance. So the margin of safety, it's a concept that was introduced in the guidance of 2012. And for a period, the margin of safety was used interchangeably with the margin of exposure. In fact, these two concepts are not really the same, because the margin of safety is basically the ratio between an health-based guidance value and the exposure estimate, in this case to the food additive. And considering that is the ratio with an health-based guidance value, the result gives you an indication of a threshold that is safe. So give you an indication of really the safety and the possible risk. The margin of exposure is instead the ratio of a reference point, so it can be an OEL or can be BMD value, and the exposure estimation to the food additive. So per se doesn't give you the indication of safety threshold. It gives the indication that there could be an adverse effect. And in the guidance is further explained that the margin of exposure can be used by the panel whenever there is evidence of the adversity, but it is not, for example, the information is not sufficient to derive an health-based guidance value. So the information of the safety is not, let's say, possible to provide. That was my last question, and I would pass the floor back to Camilla for the final wrap-up.
So thank you, Laura, and thanks to all the colleagues for having replied to the comments that we have received. So indeed, my task now is to bring this webinar to a close with a few last messages to deliver and some dates to pencil for before the finalization of this guidance document. So we remind you that so we have explained the draft output is out there. You can download it, of course, read it, and again, using the online form, submit written comments to the draft guidance. There is possibility of submitting comments until the 21st of February 2025. Then, as a follow-up of today's webinar, we will work on the presentation and the recordings to make them available on our website. So in case you know somebody who has missed the possibility of joining this webinar live, there will be still the possibility of catching up in the coming days. And finally, we mentioned earlier, we would really be glad if you could spend some minutes to fill in the satisfaction survey that will be sent to you with a link after the closure of this meeting. I think this was the last slide. So for today, I can only again thank you for joining, the colleagues for presenting the content of this draft guidance. We sincerely hope that it was useful, that we guided you through the main changes, the upcoming revisions, and that probably this webinar may have stimulated you to submit further comments for improving it until we finalize it for adoption in June. And finally, there are many ways that you can connect with EFSA, so please stay connected for future events. So thanks to all and goodbye.